This is me. My name is Sara Olsen, and I currently live here in Potsdam, New York, which is a very cold place. And this is me right now. You may be wondering how I got myself in this situation. Well, I'm originally from Binghamton, New York, and I currently live in Potsdam, New York, where I am a senior chemical engineering major. And I currently do research up there and wanted to broaden my research experience. So I decided to participate in the RIU program at the University of Florida this summer. Gainesville has certainly been an adjustment, but I've been liking it so far. When I got here this summer, they decided to pair me with the Jane Lab, led by our wonderful PI, Dr. Jane, and my PhD student mentor, Lilia. I love all of the people in the lab so much. And in the Jane Lab, we work on CRISPR-Cas research. So we'll use CRISPR-Cas9 as an example here. So a CRISPR array is a segment of DNA in bacteria that contains clustered, regularly interspaced short palindromic repeats, or CRISPR, with spacers of viral genomes from viruses that bacteria successfully defended against. The CRISPR array is transcribed into RNA that can guide the Cas, or CRISPR associated proteins, to a target sequence. This guide binds to the target sequence, and the Cas will then cut the target DNA. This is a powerful gene editing tool, as after the DNA is cut, it will be repaired imperfectly, and the gene can be turned off, or we can add a template to put a new gene in. This summer, my project is to figure out new ways for our lab to express and study proteins. I've been using DNA to make mRNA and then transfect human cells with the mRNA to get them to express our protein so we can study it. This summer, I've been using enhanced green fluorescent protein, or EGFP, as a model to test my methods. As you can see, I've been able to successfully get these human cells to glow green from the EGFP protein expression. We are hoping in the future to apply these methods to Cas proteins. This summer, I've learned lots of cool new techniques, such as bacterial cell culture, and I've furthered techniques of mine, such as human cell culture and microscopy. I've also learned how to run SCS Page and agarose gels, PCR, DNA, RNA cleanup, mRNA synthesis, transfection of human cells, transformation of bacteria, protein purification, and FPLC. It's been a pleasure to learn all these things this summer. But it hasn't all been work this whole summer. I've also had fun. So going to Universal, playing Catan with my friends, visiting St. Augustine Beach, going to the Kanapa Botanical Gardens, presenting my research at the beginning of the summer, visiting Lake Wahlberg, exploring Gainesville, going to Animal Kingdom, and seeing lots of gators on campus, getting my own UF merch and going to the World Equestrian Center, going line dancing and playing Catan with my lab, seeing Harbor live in concert, and tubing down the beautifully clear Ichetucknee Springs, going to Jacksonville Beach and visiting the butterfly rainforest where a butterfly landed on me, and going to Universal with my friends. I would like to thank everyone who made this program so great and run so smoothly. I'd like to thank everyone at the Jane Lab for being such great people to work with and such great mentors, including Dr. Jane and Lilia especially. I'd also like to thank Gabrielle, Mike, and Ashley for making sure that everything ran so smoothly and was so great in general.